Okay, the next session is the lab tour. In this part, Professor Kazuyoshi CHIBA from Natural Science Division, Faculty of Core Research, will introduce his laboratory. So he will also demonstrate an experiment using a microscope for today for us. So if you have any questions, please use Q&A box. And some of the questions will be answered directly by the professor at the end of the session. So now, Professor CHIBA, please start the lab tour.
Hello, everyone. My name is Kazuyoshi CHIBA, a professor of developmental biology, studying cell division using sea animals, starfish. So, do you know messenger RNA? Maybe you have received COVID-19 mRNA vaccine. So when your doctor administrate the vaccine, maybe he use or she use this equipment to inject the vaccine into your arms. I'm just teasing. Anyway, mRNA vaccine will make virus protein in your body. Okay? And in this laboratory, we are studying the mechanism of cell division. And in nature, starfish messenger RNAs in eggs is involved in the production of egg proteins. Okay? Not viral protein. And we construct starfish mRNA tagged with green fluorescence protein mRNA to produce green fluorescence protein labeled egg protein in egg. Okay, so before today's experiment, we have made artificial messenger RNA for study. And today I'll show you how to inject something, messenger RNA into starfish eggs. So, for vaccine, doctors use such this syringe, this big syringe, but eggs is very tiny, so you cannot use this kind of big equipment. So I'll show you how to make our very tiny equipment. So here is this machine. Using this machine, I'll show you how to make the very tiny glass pipette. So here is a glass capillary. Okay, when you add the heat and pull it, you will make a very tiny needle. I'll show you. Okay. Oh. Just a moment, please. Okay. So take a look. When I push the button, you will see the heat. Then, so here you can see the very tiny, very thin, fine needle. Can you see it? Okay, I'll take it. Okay. Oops. Here you are. Can you see it? Maybe difficult to see. But anyway, the tip is very fine. So I will use this glass to inject mRNA or oil into the egg. Because that, we modify the tip of the glass using this handmade machine. Okay, so maybe again you can see the heated wire. So using this, only 5 minutes. Okay, anyway, I will set. So I set this needle here. Okay, and this needle glass pipette is connected to this syringe. So when I add the pressure, we can inject something into the egg. So this is an egg of starfish. You can see a very big nucleus and one nucleoli. And here is a glass pipette. When I add the negative pressure, look, and when I add the positive pressure, so you can see the oil. Okay, I inject this oil into the egg. Okay, so inject. Okay, then I add the positive pressure. So you can see the big oil in the egg. So now the diameter of this egg is only 0.1 millimeter, the very tiny cells. And we can, and the volume is about 1 nanoliter. We can inject the messenger RNA. The volume of the messenger RNA is only 10 picoliter. So using this machine, we can inject a very small quantity of messenger RNA into the egg. And see, and in the next session, next video, in the next video, you will see the protein expressed in the eggs. As I told you, the protein is labeled with GFP. GFP is a green fluorescence protein having fluorescence. So my students will introduce you the distribution of protein, the green protein in the egg after hormonal stimulation using the laser microscope. Not this microscope, but another kind of microscope. And using that image, we can evaluate the protein movements or protein role during the cell division. Okay, so that's all. I hope you enjoyed my experiment. And so please turn on the video.
Okay, thank you very much, Professor Chiba. Okay, so we have a video that introducing their facilities. So now let's go on to the video.
Hi! We keep starfish here.
There are 42 seawater tanks and about 20 starfish in a tank.
This is a male starfish tank and this is a female starfish tank.
We manage seawater temperature.
Let's watch starfish. Normally, they have 5 arms.
Sometimes we find 4 or 6 armed starfish.
These are 2 feet.
They are working using 2 feet.
Mouse is here.
They eat shellfish, seaweed, and so on.
This is a male starfish. It has many sperms in the testes.
This is female starfish. It has many eggs in the ovary.
Next, microscope.
Hi, now we will introduce how we do our experiment. This is a confocal laser scanning microscope. This is based on a typical optical microscope, but instead of a lamp, a laser beam is focused onto a sample. It allows us to view tiny structures in the starfish egg that are labeled by fluorescent. Let's have a look at a movie recorded by using this microscope. This is a starfish egg. It is injected with an mRNA coding fluorescent protein. So, the localization of targeted proteins inside the egg is visualized. All of these green dots are proteins labeled by injected fluorescent. Blue dots show the egg membrane colored by fluorescent dye. Now this egg is stimulated by a hormone called 1-methyladenine. Let's see what happens. Do you see the edge of the egg is getting brighter? Let me show you again. This is before adding the hormone, and this is after the stimulation. You see, green lights get very strong, especially on the edge of the egg. This is because green labeled proteins move toward the egg surface. By adding the hormone, the localization of the protein has changed. Through our eyes, a starfish egg looks something just like a grain of sand. But using this microscope, we can clearly see dynamic changes that happen inside these small eggs.
Okay, thank you very much. Okay, now as the last part of this lab tour, so we will have a Q&A session. Professor Chiba will answer some of the questions we received. Professor Chiba, please.
Okay, so one question. Why starfish but not other creatures? Good. Thank you for the question. So actually, we use mouse or eggs sometimes, but starfish is very good for experiment because it has so many eggs in one female, and eggs are very strong. For example, in mice, we can get only 10 eggs from the female, but and also the egg is very fragile. But starfish egg is very tough, and as you can see, we can easily inject every messenger RNA into the egg. So and also another important point is that when you add the hormone to this egg, you can meiosis reinitiation is induced by the hormone. So we use starfish. Okay. And another question is that how do you differentiate between male and female starfish, do you have to do an X-ray to identify the reproductive organs? Yeah, actually, for us, it is so everyone. It is very difficult to identify the female or male. So we cut a tiny hole. We made a tiny hole in the body of the starfish and see the contents of the starfish. When you see the red organ, that is ovary, you can say, oh, this is a female starfish. When you see the white organ, that is a testis. So you can recognize this animal is male. And starfish are very strong. So a tiny organ is no problem for them. Okay, I think I answered all questions. Or yeah, that's all. Thank you. Thank you very much.